hello, everyone. Good evening or good morning, wherever you are located. My name is Daniel. I'm the Chief Operating Officer at MSAID, and I welcome you to today's webinar on our platform. I'm very happy to introduce today's program. So we're going to do a quick introduction into the topic and how we came about. We're going to talk about our newest publication, which is out in the Journal of Proteome Research, which is a scalable web-based platform for proteomic data analysis. And we will then do a quick tour of the platform, including the key feature additions that we have added since the initial release. And we will give a brief teaser what's to come for ASMS. We will have a question and answer session afterwards for those of you who have not yet been with us. Your microphones in this webinar are muted, but you can use the chat or the Q&A functionality to put down questions, and then we will pick them up during our talks, during the session, whenever there's a good time. I've also added links into the chat that cover the most important topics of this webinar, which is the MSAID platform, if you do not yet have an account for that, the publication, and also a contact point if you would like to reach out after this webinar. And with this, I would like to quickly introduce Martin, who's going to take care of the introduction and an overview about our MSAID platform, the web-based platform for proteomics. Martin is our Chief Executive Officer, and without further ado, the stage is all yours.
Thanks, Daniel. I will briefly share my screen so that we can get going on the presentation. So bear with me for a second here as I do that. You should be able to see my screen now. Can I get a thumbs up? Yes, perfect. All right, so as Daniel mentioned, I will give you a brief introduction, where we came from, where we are right now, and then Daniel is going to take over showing you actually live where we are now and giving you a bit of an outlook. But before we dive straight into why you would care about web-based processing of proteomics data, let me give you a brief history lesson on what I experienced while I was in a certain lab. For those of you, for the one listening, you know who you are. And you can do better. So yeah, here's the introduction. Basically, what I experienced was a great lab with mass spectrometry equipment that ran 24/7 as it usually does. But when it came to the analysis of the data, it really was a very manual process. So as I was very excited to get the data, I took it off the instrument and that was done using an external hard drive, and then I carried the valuable data that was the accumulation of weeks of work, months of work sometimes, and carried it to a processing PC where it was then processed with a search engine, and then I took the result of that again with an external hard drive, walked to a yet different shared compute resource, which was an analysis PC, where I did my statistical analysis, my visualizations, and then I drew conclusions from that. And all of that was very manual. And that is, in my opinion, one thing that caused me to delay projects unnecessarily because errors and mistakes happen. Over the last couple of years, we and others have worked to make this a bit more streamlined. There's now solutions where you have a computer directly attached to your mass spectrometer that does data processing. And then all you need to do is actually take your results files off that computer, store them somewhere safe, and analyze them. But this storing somewhere safe usually means, well, people throw it in their home folders and then people leave for a different position. And all of a sudden, you have to ask your colleague, oh, do you know where project XY went? I can't find the results. So that is really not a place you want to be. But there are solutions that tackle this. Albeit they are rather disconnected from the hardware where you actually acquire the data. So it's still a somewhat manual process, even if you have the first half of that workflow already figured out. But then, of course, comes data analysis, and data analysis scales with the time that your bioinformatics department can spend on this, or the experience that you yourself have. So in the end, this very long-winded but unidirectional path from raw data to actions was littered with pitfalls that I fell into multiple times, and we think you really shouldn't fall into. So an ideal solution that prevents these pitfalls would be able to deal with huge amounts of data as it rolls off these instruments, particularly newer generation instruments, can generate terabytes of data in very rapid succession, depending on how many of those you have and whether you run them 24/7. Such a system that prevents these pitfalls should not rely on local compute resources. In particular, if projects get larger and larger or get acquired over long periods of time, it should be automated and streamlined as much as possible. You shouldn't need to go to your instrument PC and carry the data manually from hard disk to hard disk, and it should allow for longitudinal and systematic data analysis. So with this sort of experience that I had, I talked about that with my colleagues at MSAID, and I wasn't alone with this experience, even though we went to several different labs around the world. So when we founded MSAID, we decided, well, at some point that has to change, and changing it will, and changing it does with the MSAID platform. So what is the MSAID platform and how does it solve these problems? Well, the MSAID platform is a SaaS platform for proteomics data analysis, and it encapsulates everything from the raw data as it rolls off the instrument all the way to data interpretation. It seamlessly scales with data volume, so it doesn't matter whether you have 1, 10, or 100 mass spectrometers in your laboratory. The system will be able to keep up with the data that these instruments churn out. And the prime reason for that is that we are using an elastic parallel compute cloud. So effectively a cloud computer, a high performance cluster that we scale up and down as necessary. As the need arises, we can add more compute resources to it. So effectively, your browser now becomes your gateway to a high throughput proteomics processing cluster without you actually needing to maintain HPC environments and knowing what Slurm even means. You can, with the system, get systematic result storage. This is built in. So as the data is analyzed, we place your results into a data lake, and there it rests and can be accessed for longitudinal data analysis over time, but also QC purposes, et cetera. You can flexibly interrogate that data either via direct API access or web access, plotting solutions, which I mentioned in a couple minutes. But you can also download the data and look at them in your environment, be that Excel or R or Python. This platform is fully API controlled and automatable, so you'll be able to get hands-free processing essentially here, all the way to your final output files. You set the experiment up once and then let the instrument acquire, and it'll process the data as it rolls in. And all of that comes with a subscription-based business model, to which Daniel will say a couple of words later on. It has the usual look of a SaaS platform. It is available, or it is accessible both on your laptop, but also on your phone if you're the type of person that wants to check whether the experiment has finished searching, from the comfort of your sofa at the end of a working week on a Sunday evening. And some of you might be. So it's all there for you to use. So without further ado, we went ahead and said, well, this is such a cool resource. We should basically tell people about this in a sort of structured fashion, which is why we put together a publication on it, which appeared recently in the special issue on software tools and resources in 2025 of the Journal of Proteome Research. So feel free to look it up. I'll highlight a couple of vignettes in this introduction about it, starting with how the platform is actually constructed. So I told you about the UI, the front end, and that it's accessible from your phone, your tablet, or your laptop. And I told you about the API server. So everything is API controlled, and that's sort of the central hub where all the communication happens. The UI and both the UI and a command line interface client interact with that API server and control functions such as uploading data, raw data that is, to the AWS S3 data lake, adding metadata to the account database or job database, what metadata we store, I'm going to go into in a little bit, communication to Stripe with respect to subscription management, but of course also, and that is the heart of everything, the scalable EC2 compute cluster, which is built on the Argo workflow engine and allows parallel processing of hundreds to thousands of raw files with Chimeras 4, as well as additional neat functionalities such as combine experiments, about which I will tell you a couple of words as well. We are using EKS here, and deployment is done via Terraform and Helm. So if you're the type of person that doesn't want to go into SaaS directly, meaning that you don't want to upload the data to some third party, but you would like to instead have that system right in your own AWS account, there's possibilities there. Please contact us if you're interested in that. Yeah. So just to give you a brief look on the sort of landing page that you will see once you make yourself an account, it is structured into a couple of key pieces of information and important metrics, such as, for example, the number of uploaded raw files, the number of FASTA files that you have, the number of experiments, and these are searches of one or more raw files, the number of running experiments currently, as well as a couple of information pieces on user management, visualizations, etc., etc. Daniel will show you all of this in much more detail when he's doing the live demo. But of course, you will also be able to take a look at the resources that are available to you. So how much storage you have left in your account and how much compute you have left in your account. I told you that this is a SaaS platform, so eventually we need to get data into it, right? So how do we do this? Well, you can do this directly from your web browser. So effectively upload data from locally accessible file systems, be that either on the computer with which you're accessing the website or on a mounted drive such as a NAS, SAN, or some other storage appliance, directly via the web browser to the data lake for it to be available in the platform. And that is extremely fast. So we put a lot of effort into maxing out data pipes so that you don't have to worry, oh, I have this big experiment. Will it take forever to upload that data? No, it won't. The theoretical upload limit on the line that we tested, which was a gigabit connection, was 80 seconds for a 10 gigabyte file, which is 128 megabytes per second, or a gigabit per second. And when you're using the command line client, we come very close to this theoretical limit. So here it takes 97 seconds to upload this 10 gigabyte file to the platform. When you do that through the browser, you have a little bit of overhead that you need to deal with. So that time goes up to 162 seconds, but it's far less time than would be required to generate these 10 gigabytes of data. So you don't have to be afraid that this is gonna be a problem in the long run. I mentioned a bit about metadata because the metadata is something that everyone is interested in when it comes to contextualizing your analyses. Metadata in the platform are stored as tags. So to each and every raw file, you can add tags that describe what that raw file is. So for example, here we have tags such as HFX, denoting that this raw file was acquired on an HFX. It's a TMT Pro raw file, so we added that tag too. We have a couple of PXD numbers here, maybe the author of a publication or the PTM. And in the future, what we envision is that we are able to auto-generate some of these. So for example, the tag HFX. This is, of course, extractable from the raw data, but also other metrics such as, for example, retention time could be extracted from there. So we are currently working on a system to effectively extract interesting metadata information directly from the raw file and make that available as tags here. And how do tags help you? Well, tags help you to locate files of interest and to also then later on use particular files as conditions, for example, in your analyses when you do contrasts of treatment versus control, for example. Tags are editable for each raw file, for each FASTA file, for each experiment. You can add as many tags as you want. These are free-form tags, so it's just text-based elements here. But if you add a new tag, we check our database whether this appears already in our database so that you don't generate redundant metadata that might make it hard later on to find the HeLa cell spelled with a capital H or with a lowercase h. I mentioned the parallel compute cluster and that it is currently being driven by Chimeras 4. This is the first search engine that we integrate in the platform, but the platform is built in such a way that it doesn't require to run on Chimeras only. Of course, we're the developers of Chimeras and we love Chimeras for what it is, but this is for you interested parties out there that might be interested in integrating with our platform. It is fairly easy to incorporate other search engines here. So if you have interest in your search engine to be incorporated into that platform, please contact us. A couple of adapters need to be written to, get the data in the correct format. But in principle, this platform is independent of the search engine on which it runs. When it comes to experiments, I mentioned an experiment is essentially a search of one or multiple raw files. So here we have 2 experiments, for example. You see that the first one has 6 raw files, the next one has 10 raw files. And for the next one, we actually are looking at a little bit of a detailed overview of all the information contained in this particular experiment here. This is to show what type of metadata and additional information we actually store and display to you in a convenient way. So we show you immediately, was this run successful or not? What did it maybe have a couple of errors in some raw files? We show you the name of the experiment, of course, how many raw files are there, when it was created, by whom, how long it took to run it, how much compute we charge, but also high-level statistics on the number of identifications, such as, for example, here, the number of PSMs, precursors, etc., as well as tags. You can also check the actual current status of the search by looking at the log messages. There, effectively, the log of the search engine is updated in real time. So if you're sitting on the sofa enjoying Tatort on a Sunday evening, you can check whether the search ran through completely, whether it's through a couple of errors or warnings, or whether you can dive straight into data analysis first thing Monday morning. The CLI client is another point I briefly wanted to touch upon. I mentioned the distinction between uploading data via the web interface or via the command line client. The command line client, while it might seem daunting for say non-bioinformaticians, it is actually fairly straightforward to use because it wraps essential functionality of the platform in very easy to understand commands. It is available for Windows, Linux, and Mac OS. Normal authentication with the web platform runs via AWS Cognito, so you use your same credentials that you use for logging into the platform in the web browser, also for accessing the platform via the CLI. And a couple of vignettes for that CLI client are, of course, you can upload data. But what I find particularly useful is the watch function for folders. So essentially, you tell the command line client, hey, this is a folder where in the future there will be raw files. Once there is a raw file there and it is completely there, finished acquiring or finished copying, please upload it directly to the platform, search it with these settings, and download the results into this folder. So it can completely automate everything from the finish of data acquisition all the way to you being able to look at lists of identified proteins, peptides, etc. But you can also use it to upload the raw data, search it, and don't download the results yet, because once they are uploaded and searched, what you can take advantage of is the combine experiments functionality, which I think is very powerful because it allows you to process a big longitudinal study that you acquire over maybe multiple months as the data rolls in. And once the final raw file is acquired, you can actually process the files immediately without having to research every single raw file. So what does that mean? If you would want to set something like this up, you would set the command line client to watch a particular folder where you know all data from a particular project is being deposited. Then the command line client will take care of uploading that data to the platform, searching it with a config that you give it. You can configure this config in the web interface and then download it to use it with the command line client. And then the data would be searched raw file by raw file. But of course, you want to take advantage of matching between runs functionality, right? And that is possible with the platform. So even though these files were searched individually as they rolled off the instrument, at the very end, you can click this Combine Experiment button here, choose the experiments that you want to combine, and all we are doing then is sort of what we call post-processing steps. So roll up of FDR to the global level, calculate protein groups, protein group FDR, quantification, including match between runs. All of that is done, but not the search. And the search is what actually takes the most time. So we think this is a very nice feature for those of you that have these very long-running experiments and want to analyze the data as soon as the last raw file was acquired. We have a sort of weird-looking figure in the publication that when I first saw it, I laughed a bit, and that is just comparing, how do the results of multiple raw files that were either searched directly together, so we uploaded them all together, we searched them together, and then looked at the data, compare to the results of the same data if we later on combined them. So for us, it is identical because we're doing the exact same things when you combine the experiments later, as opposed to you search the raw files in one experiment from the get-go. And I think that's actually very powerful. So you can be sure that it doesn't really matter whether you search all raw files in one experiment or combine the raw files later on, you will get the exact same results, which I think is pretty cool. All right, so what does it give you in terms of processing speed? Well, if you are the type of person that wants to process a raw file as it rolls in, then you would be here on the left-hand side. So you would be uploading one raw file and then processing it, and then once the next raw file is acquired, an hour later, you would process it again. And that is entirely possible. The system will be able to keep up, even if you're searching, say, immunopeptidomics data here, which of course takes longer to search, the system would still be able to keep up because we parallelize searches in the cloud. But if you're the type of person who likes to search big experiments in one go, you can actually take advantage of our scalable EC2 compute cluster here, where we have hundreds of CPU and GPU cores available on demand to parallelize your searches. This is just a plot of how long it takes to search one raw file, 10, 100, or 500 raw files on the system. And you can see that here searching 500 raw files takes 302 minutes. And that is, while this is 500 more raw files, it is not 500 times longer than searching a single raw file. And that is, of course, because we parallelize, and we parallelize currently 100 files. So 100 files are processed in parallel, meaning that with 500 raw files, you end up waiting for 302 minutes, and you'll have the results of these 500 files. This is just an example. We have done much bigger experiments where we searched 5,000 raw files together already in one experiment. And we have a recent pilot study where we searched 26,000 raw files in a matter of a couple of days. So the platform really is scalable to population scale plasma proteomic studies if you so desire. So just, I'm a bit cautious about time. So because Daniel is going to show some of that functionality, I will not go into too much detail here. Just suffice it to say that on the data exploration side, we offer a so-called data browser where you can look into the tabular output files of your searches, meaning you could inspect, is my favorite protein there? How many peptides did I identify it with? And what are the precursors that map to these peptides? One recent addition is actually the precursor quantification trace, where you can actually look at the quantification of a given precursor based on our Chimeras coefficients. And this effectively visualizes how the chromatographic peak shape of that precursor looked in this particular raw file. A couple of other visualizations that you can use on the platform are bar graphs, scatter plots, volcano plots, etc. And they all come together in a sort of in a dashboard kind of fashion. Just to give you a couple of examples, the visualization dashboard here on the right-hand side has a typical volcano plot, just a bar graph of the number of peptides identified per raw file, a principal component analysis of the different files. We also support upset plots, as well as a couple of other cool graphs that will be available soon, i.e. distribution plots, QC plots, etc., etc. And if this is not enough for you, as I mentioned, you can download the data from the platform in long and wide format, and then either hand it to your bioinformatician or open R yourself and plot these plots by yourself. We will provide an R package to directly contact our backend data lake, so that you don't even have to use the command line client anymore as well. That is planned for ASMS. Right, and with that, I am basically at the end, and all there is to say from my side is, please just give it a try. You can create a free account at platform.msaid.io. You need to enter your email, password. Please be aware of the password rules here below. Just a couple of precautions to not have too weak passwords. You then accept the end user license agreement, and boom, you have a free demo account, which allows you to process 100 gigabytes worth of raw data in order to evaluate the platform and see whether it's a good fit for you. And with that, I'm at the end of my part, and I would be open for questions if there are any. But we can also move to the live demo and postpone questions till the end if that is preferred. Thank you very much. Looking at the Q&A, I don't see questions yet. I don't see questions in the chat either. So I'd say, Daniel, maybe you can take it from here.
All right. Thank you so much, Martin. Thank you for this overview. And I will dive into giving you a couple of live interactions with the platform and explain a little bit about our business model, about our subscription model, about all the nitty-gritty details we have not yet been talking about in the rather high-level overview. So when you go to the platform at MSAID.io, you need to log in and you will be presented with our homepage. And as Martin said, it captures basically all the necessary informations that you have when you look at your data sets, when you look at your experiments. It has a classic software as a service setup, meaning that you'll have your navigation on the left-hand side and you'll have your user account on the right-hand side, including several options that I will go into. We also have created a product tour, meaning that if you do not want to listen to me, but rather would like a computer to take you around the platform, you can just do that and you can restart this tour at any time point on our panel. So before I talk about all of these panels and about how to start an experiment and how that looks like live, I would like to highlight a couple of things. So since recently we have introduced two-factor authentication for the platform, so far we have been using AWS Cognito, and that now has been extended to also be compatible with two-factor authentication, meaning that your connection to the platform is even more secure. And with that, I would like to directly jump into our subscription and payment model. So as Martin explained, at the moment, this is a software as a service offering that we host as MSAID. It is hosted in Germany. It's fully GDPR compliant, and I'll have a couple of more points towards compliance in one of the later slides I'm going to show in our Outlook slide deck. But how this works is basically that here on the right-hand side, you see that you will have resources, meaning they have storage and compute. And it's basically like a phone plan. You have different tiers that you can connect to. And when you buy these tiers, you have a certain amount of storage, you have a certain amount of compute, and that will refill monthly on a monthly basis. So when you register for the platform, first what's going to happen is you're going to be put on a trial plan that's going to run for 30 days and has a certain amount of compute included. In this case, you can process up to 100 gigabytes of data on us. If you really need a lot more to try things out, talk to us. We have been running demo experiments. We have been running larger studies with some of the pilot customers in order to convince them of the platform. When you then go to the subscription tab, what you can see is what your current plan is here, and you will see that we have different tiers depending on your needs. The different tiers that we have here are all different in the number of compute and the number of storage that is included with them. We have a starter plan that you can use to get a first grasp on the cameras platform, the MSAID platform and cameras. And we have a premium plan for people who want to run more than one mass spectrometer all the time with all the data coming off the mass spectrometer to be processed on the platform. Obviously, we can tailor an enterprise plan to your needs depending what you require. You will see that we offer yearly and monthly subscriptions, and you can actually check out, and this is one of the new features that we have introduced recently, you can check out that feature without having to talk to us. You can basically just buy the plan and we'll connect you to a Stripe session where you can use a credit card or other payment methods depending on your location to directly buy that starter plan, and that will be active immediately. For academic customers, we offer a rebate program for these rebate programs up to 30%, please request a quote. Click any of these buttons to request a quote, and we'll get back to you within 3 business days and try to talk to you. What is your desired setup that you'd like to have? What amount of data you want to run? What is the background? What kind of institute are you? Can we maybe help you with publishing a paper? And can we help you getting a talk on ASMS? And all of these things are factored in when we have these chats about how we can tailor a quote for the academic researchers. If your compute and your storage is not enough, you will also be able to buy one-time compute upgrades. I'm currently on a free trial plan here for this account, so I can't purchase this compute update. But once you have one of our plans, you can just basically purchase one-time upgrades that you can use if you need more compute than you have in your account. You can just, again, comparing this to a phone plan, if you go somewhere and you want to stream a large game, or you want to stream a lot of Netflix, you might as well get another big data package this month in order to do that. And once you have purchased all of this plan, you don't have to use it alone. The platform has built-in user management, which allows you to invite users and collaborators to look at the same data without having to create a lot of different accounts for accounting reasons, which usually drives the finance department nuts if you buy multiple accounts for people in the same lab. So what you can do is you can just basically buy your bigger subscription, invite your coworkers. You will be able to collaborate on data. You will be able to process data together. You will be able to see and share the same visualization dashboards and results, all with one account owner who is our person to talk to in terms of billings and the administration part. But now let's get back and process some data. As you can see on the right-hand side, the most important tabs are your files and your experiments. And when we go to the Your File tab, you will see that I already uploaded a couple of raw files, which you can see in this table view. The table view can obviously be sorted in any way, shape, or form. You can add various filters if you want to build bigger filters, or you have lots and lots of data uploaded. And you will see that here are our free field tags. You can use these tags also to subset your results and your data, so you can find back all the raw files that we're looking for in a easy and simple fashion. On the other hand, you obviously need to upload some FASTA files. So I uploaded FASTA files here. You can set the parse rules for these FASTA files so the proteins get properly parsed out of the FASTA that you're using and are available for later processing. I'm not going to show you how to upload files. I'm going to rather show you how to start an experiment. You'll see on the experiment list that there are a couple of experiments that we already created, but we're just going to create one more to show you how the process works. We're just going to create an example HeLa run here. We can set additional tags. I can, for example, add the tag webinar here, or I can create a new one. And then I'll have to select my algorithm. Chimeras is always available in various different versions. You can select the latest version, but we're also going to keep older versions of the algorithm around for you to go back and process data with a version that you had before. This is especially important if you have studies that run for a longer period of time, or if you had to rerun a couple of older data sets. In this moment, Chimeras 4.3 is the latest version, but we are planning to put a beta version of Chimeras 5 on the platform very soon, before ASMS, for people to test. So once we have selected our algorithm, it's pretty much straightforward. We select a FASTA file, we select one of the raw files. In this case, I'm just going to go and look for my HeLa file. I'm going to see I have a HeLa file here. I'm just going to use that. And then I have to go through some necessary configurations. But in order to not go through these necessary configurations all the time, we also have a template selection, meaning that if you don't want to set your templates, your settings, you can just save them once and then directly jump to submitting the experiment. If you have not yet set these, you can change your selection of all the usual parameters, the enzyme that you're using, the variable modifications that are using, and we're by now also supporting quite a lot of modifications including TMT, TMT Pro, phosphorylation, and ubiquitination acetylation. We'll be adding quite some more this ASMS. As for quantification, Chimeris has a built-in deconvolution version of an MS1 quantification, including match between runs, meaning that we quantify in MS1 just as we do in MS2. We deconvolute the MS1 spectrum and ask the question whether how can we best match the isotope envelopes of peptides into the MS1 spectrum present. Obviously, we also have an MS2-based quantification that is just that coefficient from our deconvolution function that asks the question, how much of the intensity is actually explained by the peptide that I have at hand. We also support TMT MS2 reporter ion quantification. MS3 reporter ion quantification is something that's going to be added relatively soon. The quantinull file option or the MBR option is basically what you know from MaxQuant, taking into account information from other raw files that actually identified an analyte at that very position. And we are using this information even though it is not making the FDR threshold in this individual file. Another thing that I want to highlight is that there is an experimental design where you can either upload an experimental design via CSV or specify it. This is especially important or useful if you want to do some of the downstream analysis in the platform itself. So you can, for example, enter a sample name. Here, I only have one sample, so that's pretty boring. But if I would have multiple samples in here or even fractionated samples, I can put all of that down right here to carry this meta information through to the end. And with that, I get to a summary page where I can quickly review my search. I can download the settings for the CLI client, meaning that I don't have to engage into building custom JSON files. I can just click them together and download them and use them for my runs that I want to do with a CLI. If you, for example, want to run a phosphorylation search, you can basically just configure it just as you want it, download the JSON, and you don't have to worry about submitting illegal settings or having to worry about breaking the JSON schema when you manually configure something. You can also save that as a template, or we can just add the experiment to queue. In the meantime, on a different screen that you can't see, I also just submitted another small experiment via our CLI client. You can see that our run here is now going into the running stage, and here is my auto-generated name for my CLI run that I just ran on a different screen. When you look at the experiment, you will be able to see the log of the files. Let's go to one of the experiments that have already been successfully created. You will see high-level information about proteins to PSM levels. You can see the experimental files that were used. You can see the experimental settings and download these, and you can also resubmit them with a click of a button if you want to change settings or if you want to resubmit that with a newest version. This is also one of the usability features that we have recently added. After your searches have finished, what you can do is you can explore the data directly in the platform. And I just want to show that here really quickly using the data browser. The data browser is basically a in-web version of Excel, looking at all of the identifications, looking at the different peptides and precursors in a nested fashion. But I just want to highlight what Martin already showed before, that there is the possibility to now look at the quantification traces for these individual experiments. And when I click here, I see the actual identification trace for this peptide and the MS1 trace, how we quantified that over retention time. And you see that we, in this example, had quite a bit of tailing, and the chromatography is not as pretty as it should be. Still, this is a great way to first look into your data. You can build rather complex filters if you would like to do so. And for example, look if your favorite protein is here. And the next step then would be to look at some of the results. Therefore, you can go to these visualization dashboards that is shared between users and that is also saved. In this case, I have a astral study with 3 samples from a wild type and 3 samples from a knockout of a certain gene, and I can quickly generate a volcano plot that is interactive, that can be zoomed, that has a pop-over function, and where I can download all the data and also export the underlying data if I want to have this for submission. All of this is reactive, meaning that you can also just move around and export these files. And we imagine that by the time we have added a lot more visualizations, this will be actually pretty useful for a quick glance into the data, also for QC reasons and others. You can just add a plot by, for example, selecting the precursor level. We're adding a bar plot per file. We click a button. We want to have this in green, and we create the plot, and then this should show up in a second. In the interest of time, I will now jump back and show one more feature, which is the download that we have quite drastically refactored in the last couple of weeks because users requested that. So now in the download functionality, you can select a long or a wide format. The long format is what you would use if you, for example, want to plot stuff in R or Python. It would mean that every single line is a protein for in a raw file in a certain condition. The wide format is what people would like to use if you look at data in Excel, where every column should be a sample and your rows should be proteins, and you can with one simple look see the different quantifications across samples, for example. You can select different levels you want to export. You can also select all unfiltered data. And once you request that download will get prepared and is available for you to download after a couple of seconds. With that, I would like to stop the demo in the web interface, and we'll take a couple of 2 minutes to go over my last 3 slides before we go into a Q&A session. For that, let me quickly change my share. All right, and we should be back with our slide deck. And I want to spend a couple more minutes on the outlook, what is to come. ASMS is around the corner, and all of you know that ASMS is one of the major conferences. Hence, we also sat down and put together a couple of important features that we will release this year. I can't tell you too much because obviously you shouldn't publish your stuff before ASMS, but a couple of teaser slides will be there. So one of the main focuses of this year will be to make high throughput proteomics possible. We have spent quite some time on working on our scaling efforts, pushing our workflow manager to its limits in order to allow the processing of thousands of files in parallel. At the moment, we have been working on single pile performance through clever CPU scheduling, meaning every single run will benefit from that. We are now allowing up to 500 raw files processed in parallel. We have implemented significant performance gains for multi-file processing, especially when we have to collect all the data for protein rollup and FDR. And we have been running scaling up experiments of up to 5,000 raw files in a single experiment and 26,000 raw files in a larger study across multiple experiments in order to prove that this is actually something you can do in quite a short period of time. In the future, we will see a lot more efforts into this direction because with all the data amounts that we generate by now, it will become necessary to move all of these processing pipelines into the cloud. At least that is our standpoint here. We've also heard customers and heard the requests in order to cut red tape when applying our software in pharma environments, in biotech environments. This is why over the last couple of months, we have built a state-of-the-art information security management system and have gotten this certified by the TÜV. We have successfully survived, I might say, 2 audits in the last 2 months, and the certification process is going to be completed in April 2025, meaning at ASMS, we will be an ISO 27001 certified organization. This usually helps a lot when trying to explain to your IT department that you're trying to upload data to some point. And we're very happy to talk to you and your compliance team if further needs should arise. We've also added a GDPR compliant data processing agreement as default attachment into our OILA, and we'll update our OILA in May 2025. This again helps a lot when dealing with compliance compartments if they want to see technical and operative measures, how the data is kept safe, and if they are afraid that the mass spectrometry data is containing personalized identifying information, for example, in file paths, in sample names. With this data processing agreement in place, that is way easier for compliance people to accept data transfer to the cloud and will also help you make a case with these departments. We're looking forward to meeting you at ASMS in Baltimore. As I said, we will have quite some presence there. We'll have a booth, 2 or 3, that we will send staff over. Martin's going to be present. I'm going to be present. I'm very happy to meet you in person if you would like to discuss about any of these topics or if you'd like to know more. We're going to be celebrating also our launch of Chimeras 5 in our breakfast seminar on Tuesday, June 3rd. We're going to have more updates to the MSAID platform. As I said, we're working on our prototypes for an R package directly interfacing with this. And we're also excited to have scientific speakers that are actually using the MSAID platform for their studies in this breakfast seminar as well. Also, we were able and very, very happy to score 3 talks and 2 poster presentations during the scientific program, where we will share more insights about Chimeras 5, about new deep learning models, and about our thoughts on error control, and also some new compatibility updates. As a mini teaser, we will launch Chimeras 5. We will show across the board improvements in peptide and protein identifications. We will also be showcasing in Ferris 5, where we are slowly but surely moving into an all PTM model fashion with added PTM support for larger modifications, for various other modifications. And we believe that this is quite a jump in our deep learning capabilities that we're going to showcase there. And also, and this has been long awaited for, we will be introducing compatibility with Bruker-Timstorff data, meaning that it's not only thermo raw files that you can process on the platform, and we'll hope that we'll have a beta version of that online relatively soon for people to test and give feedback. And with that, I would like to end my quick overview of the platform and what's to come in the future. And Martin and I are very happy to take questions. Maybe Martin, because you have been monitoring the chat and the Q&A session, maybe you can already start maybe paraphrasing and summarizing a couple of the questions that have already been asked, and I will jump into that in a second.
Sure. First of all, thank you very much to all of you for attending today's webinar. We're going to start the Q&A. The first question that was asked is whether our software is capable of analyzing DA data and whether it can analyze many modifications, including phospho, UV, etc. And to that, I can say absolutely yes. Chimeras is capable of analyzing DDA, DIA, and PRM data, and supports, among others, phosphorylation, ubiquitination, acetylation, Silac, TMT, you name it. The list goes on. Please check out MSAID.de/Chimeras for more information on compatibility. But yes, absolutely. This platform is meant to be a unifying platform for the processing of bottom-up proteomics data. If you want, you can also check out our bioRxiv preprint. We are in very late stages with this paper that is unifying the analysis of bottom-up proteomics data with Chimeras. I'm going to take a minute to find the DOI identifier and paste that in the chat as well for you to find. So if you want to have a read of that, feel free to do so.
Thanks, Martin. If there are any more questions, feel free to put them into the chat or feel free to put them into the Q&A functionality here. I also would like to say thank you for everyone taking your time so far. I can just invite you to give the platform itself a spin or reach out to us. We're very, very interested in learning, understanding, and working with you to make that processing pipeline part of your data journey. And we are very happy to hand out demos to people that are interested in that. And we are working together with pilot customers that have a couple of more non-standard use cases to see whether we can make these use cases working for you. As Martin also said, at the moment, this is a SaaS platform hosted by us. On the other hand, there is no reason why this platform should not run in any other AWS account or in your organization. That means if there are constraints by your compliance department that this is something that you need to have in your account, please reach out to us. We can change from a SaaS model to a licensing model where you would basically get one of these copies. The only thing we would need is to start discussing what exactly are the concerns and constraints, because that has been proven to be very individual and cannot be generally stated. I see one more person typing, and I guess with that question would be whether there is normalization for a set of multiple files, and is there an option to do by selected proteins in a FASTA instead of by all? So as for normalizations, the data that we're reporting at the moment is raw output data as we detected in the mass spectrometer, meaning that if you want to apply LFQ kind of normalizations or other median centering normalizations, you would have to do this in your post-processing. However, we have also heard requests for building these functionalities into the platform directly, and there are work streams where we are adding different roll-up methods, for example, selecting how do you want to deal with shared proteins, how do you want to normalize data. So there's going to be features to come in this regard.
And then there's another question. How does Chimeras handle chimeric spectra differently from other traditional search engines? And if we could give an example on how this has impacted sensitivity or ID rates in complex samples like plasma. Here I can absolutely point you to the Chimeras preprint because there's an in-depth description of how the deconvolution of that is employed by Chimeras actually works, and you have comparison to other search engines. But in a nutshell, what we are doing is we are trying to explain as much experimentally observed MS2 fragment ion intensity with as few peptides as possible. In mathematical terms, we are performing a non-negative regularized linear regression, and that means that we minimize the number of peptides that we use in order to explain as much experimental intensity as possible. So why is that advantageous? When we compare this to other traditional methods of identifying more than one peptide from a given scan? Well, the other traditional methods are what we call the subtraction and duplication approach. The subtraction approach essentially removes peaks that were assigned to a particular peptide and then resubmits the scan again. And the multiplication approach just duplicates the entire experimental spectrum for each precursor detected in the isolation window and submits these scans separately to the search engine. Both have the disadvantage that they either use data not often enough in the case of shared fragment ions, or use data too often. So you can imagine the Y1 is a fragment ion that is very frequently shared by multiple peptides that are co-isolated at the same time. Chimeras will proportionally distribute the intensity of that Y1 ion to the different peptides given their overall intensity in the scan. And that has the added bonus that we make the best balance between sensitivity and specificity and can maintain accurate FDR control. It is entirely spectrum centric. So for each and every scan, we identify a peptide and we get a quantity for that peptide. The quantification in chimeras effectively means what is the contribution of peptide A in scan 1? What is the contribution of peptide A to scan 2, to scan 3, and so on? And these contributions we then plot across retention time. And this is what Daniel actually showed in the quantification trace. This is exactly the value that we are using. And this is spectrum centric, fully spectrum centric. We then, of course, aggregate it over multiple scans to calculate what we call a pseudo extracted ion current to quantify a peptide. But that's effectively it. On the back of chimeras, we can dig substantially deeper into highly complex samples. We showcase plasma proteome analyses in the paper that I mentioned as well. So please, like for the nitty-gritty details, check out the preprint. You'll be happy about the detail that we put into this. All right, another question is, will Chimeras 5 be integrated into Proteome Discoverer? We are currently working towards this. So Daniel has answered that question. And then, yes, I don't see any other question on the chat side. Is there a question on the Q&A side? No.
All right, I guess then with that and 2 minutes after the time, I would slowly but surely close this seminar. However, I know that most of the questions usually pop up the moment you leave the chat or you leave the window. So if there's anything that is still unclear to you guys, please reach out. We're very happy to walk you through individual parts or do another Q&A session in case these topics are of wider interest. So with that, thank you very much for coming today. Thank you very much for your time. We really appreciate that. And we are very much looking forward to seeing you in Baltimore. So if you're there, please make sure to stop by the booth. And we are happy to talk there.
Yep.
With that,
thank you very much.
Thank you and goodbye from Germany. And I wish you a very nice evening if it's not just the very morning for you guys and you still have to work.